Hello, lab class. Welcome to the first pre-lab video about the first lab that we're going to do in Biochem lab. This is going to be our titration of amino acids lab. So first, let's talk a little bit about the structure of an amino acid. The amino acid has three sort of main parts. There's the amino group, the carboxylic acid group, and the side chain, which is represented by the letter R. And when these groups are in solution, depending on the pH of that solution, each of these three groups has the potential of being ionized or protonated and deprotonated. So first let's start with the amino group. So the way it's drawn out here, you have a central nitrogen with two protons, two hydrogens, attachment to the rest of the molecule, and then there's a lone pair of electrons. However, in some cases, the lone pair can gain a proton, and this would be in a low pH environment where there are a lot of protons in the environment that are sort of there and available to protonate this nitrogen. And in this case, the nitrogen would actually gain a positive charge. At higher, and that would be called protonated. At higher pHs, like we see drawn above here, we are deprotonated, and there's no charge on the amino group. So on the other side of the amino acid, we have our carboxylic acid group, which has our carboxylic acid, which is attached to the rest of the molecule. And that also has two forms, the carboxylic acid form and the carboxylate form. So the carboxylate form is deprotonated, which you would see at a high pH where there's fewer protons in the environment, versus the one drawn on the top, which you would see at a low pH. This is the protonated form. And in this case, the protonated form is not charged, and the deprotonated form is charged. Whoops. My pen is acting up today. All right. And that has a negative charge. And the last group to consider here is the side chain, which is drawn by the letter R. Some amino acids have an ionizable side chain, and some of them don't. And you'll see, as I explain later on in this video, that can make it easier for you to identify which amino acid you're working with. So the one last thing we should think about here is what does it mean when I talk about low pH and high pH for the difference of when the parts of the molecule are protonated or deprotonated. So as we discussed a little bit in the chapter 2 videos, the pH, the point of the change is the pKa. So a low pH is going to mean below the pKa. A high pH is above the pKa. And that's true. And the amino group has its own pKa. The carboxylic acid group has its own pKa. And for the ionizable side chains, they can add a third pKa. And yeah, so let's go on to the next slide to talk about what this means in terms of what's happening in our lab. So this next slide shows our experimental setup. And you'll see we have a ring stand over here with a clamp that's holding onto this burette, and the burette has volumes marked along its length. You can fill up the burette with a funnel. And in the case of the titrations we'll be doing in class for this lab, we're going to use two different, one acid and one base, hydrochloric acid and sodium hydroxide, to fill up these burettes. At the bottom of the burette is a stopcock, and this allows you to add the liquid dropwise into this beaker, which is filled with a solution containing either your amino acid or the control solution. And it's important while you're filling this dropwise that there are not any bubbles in your burette that can interfere with dropping the liquid into the beaker. So as you fill your burette, if you see bubbles, you can gently tap the burette to try to disperse them, or you can dispense out some of the liquid to try to move around some of those bubbles. And the other item that's inside of our beaker is the probe for our pH meter, which is drawn to the side here. And the whole thing is on top of a stir plate, which has a stir, and the beaker has a stir bar in it that's gently stirring the solution to keep everything uniform as the pH is measured. So as you go through with this experiment, you will add the acid or the base dropwise, and you'll measure pH changes. If you choose to work with a partner, it might be convenient for one of you to read the pH from the pH meter and the other one of you to read the readings on the burette. But I highly recommend that at some point during the experiment you switch and have both partners reading from the burette. We have enough spaces in the lab this year that you can also choose to work as an individual. That's up to you. So a couple things to note about reading the pH meter. So as you read the readings, so first of all, you'll have to, before you begin the experiment, you'll have to calibrate the pH meter. So when we're titrating with an acid, you're going to calibrate using two standards at pH 4 and 7. When you're calibrating with the base, you're going to calibrate with two other standards, one at 7 and then one at 10. And this is because you want the standards that you calibrate with to be similar to the pHs that you're actually wanting to measure. So for acids, you want a low pH. For bases, you want a higher pH. And all of this information is in the procedure document that you should have. And the other important thing to note when you're reading off of your pH meter is that as you go through and you're adding your acid or your base dropwise, sometimes if it's been a long time since you've added a drop, or if for the reading the pH stays sort of steady for a long time, the pH meter will sort of stop reading and think that you're at a stable pH and it's done. So it's very important when that happens that you press the read button again. Otherwise, as you keep adding acid or base, you won't see a change displayed on the pH meter display. And the way you can tell if you're measuring or if it's sort of done reading and stable, is there's a little A drawn on the display of the pH meter that's showing that it's reading. And when it's stable and it stops reading, it will draw a little line over the top of it. So please keep an eye out for that, and if you do see it, it's not a problem. Just make sure you press the read button again before you continue. So that brings us to a little bit more about what you should know about reading from a burette. So on the left side of this diagram, we have a sort of cartoon diagram, and we can see the burette, and it's marked in volumes. The volumes increase as you go down because you're measuring the volume that is dispensed. So as the liquid falls down, you've dispensed more volume. And there are also markings on the burette. There are numbers marked every milliliter, and there are also markings for every 1/10 of a milliliter. So for each of these lines, you have 32 milliliters, 32.1, 32.2, and 32.3. And you might notice that this liquid, because it's in a narrow container and because it's an aqueous solution, you have this meniscus that forms on the top. So whenever you're reading a volume, especially a really accurate volume from a burette, it's very important that you read from the bottom of the meniscus. So if I were trying to read from this drawn burette on the left, I would imagine a little line going across the bottom, sort of at the bottom of the meniscus. And here's where we have to consider how many significant figures we're going to read. So you're already given these 3 significant figures just from the burette, and we already know that. And we can also have one extra digit as the uncertain digit. So you have 3 certain digits and one last uncertain digit. So in this case, it's pretty clear that we're somewhere between 32.2 and 32.3. but we're going to add one more. So to me, to my eye, this looks like it's about 32.6. Sorry, 32.26 milliliters. And it's okay for that last digit to have some uncertainty. Maybe it's 32.25, maybe it's 32.26. But we know for sure it's not 32.21 or 32.28. So that's why we want to add in that extra uncertain digit at the end. On the right, we can also see a diagram that shows a real-life picture of your burette, so it's a little bit harder to read. But again, you can sort of see, change the color again, it's hard to look at. So you can see that the bottom of the meniscus sort of falls right along here, and conveniently it seems to be right on this line. So in this case, we're counting down, so 42.0123, so we have 42.3, and then we add one extra uncertain digit, so you just add a 0 on there, so 42.30 milliliters. So now that you know how to read a burette, we're ready to talk about drawing a titration curve. So as you add your acid and your base, you can start to think about what that would look like for your curve. So in the middle is what your solution would look like with no acid or base added, and then you would titrate down to the left with an acid and over to the right with a base. That's on your x-axis is how much volume you've added of your titrant, and which is important. Which is why it's important to measure out the volume that you've dispensed. And I also want to make one other note, actually from a couple slides back. I apologize for forgetting this, but when you're dispensing out your titrant, you're dispensing it drop by drop, and you're looking for pH changes. You're making recordings of the pH change about every 1/2 of a pH unit. So sometimes, depending on this titration curve, which we're just about to talk about, adding one drop might change the pH a lot. It might change it even more than 0.5. That's okay. There's sort of nothing you can do about that. Just record whatever pH you got after you got that drop, and you can add that into your graph. Sometimes you'll add multiple drops of your titrant and you won't see a big change in your pH, and that's okay too. But so you can just measure then the volume that you've dispensed every 1/2 of a pH unit, or as close as you can get. Which brings us to this curve. So you can see there are flat parts of this curve and there are steep parts of this curve. So you can sort of imagine when you're adding your titrant, you're moving down to the left or over to the right. And so if you're sort of at this 0 point, you can see that the pH drops pretty dramatically with a small amount of change in the volume. So you might add a drop and see a big drop in pH. Over here in the buffer zone, you would see a much less dramatic drop in pH. So if you add, 1 or 2 even mils of acid here, you don't see a big pH change. And this is because you have a mixture of both the protonated and the deprotonated form of your amino acid, and it is acting just like a buffer would, where you have the two different forms that can add and subtract protons as needed to kind of buffer against changes in pH. The steeper points of our curve are what are called equivalence points. So right here in the middle, the steep point, we have our equivalence point. And we have another steep one right over here. So this is our second equivalence point. And each one of those represents when your amino acid has fully, depending on which direction you're going, fully lost or fully gained all of the protons. So it's going to very quickly change pH because you no longer have a mixture of the protonated and deprotonated form. You only have one form or the other, and that causes that really dramatic change in pH. Halfway between the equivalence points, we have another important marker that we should note. So these are our half equivalence points, and those are at those flat regions, the buffer regions. And And these are where, like we said, we have a mixture of our protonated and deprotonated forms. And this is also known as the pKa, which you should hopefully remember from when we talked about pKa's in Chapter 2. So the pKa is that midpoint where you have a mixture of the two species, and that's at halfway between the equivalence points. So when you measure the volume of titrant that you add and the pH, you can draw a graph like this, and that will help you determine the equivalence points and the pKa's. And you can use the number of equivalence points that protonating and deprotonating that we talked about on the first slide to figure out how many ionizable groups you have. So we already talked about how every amino acid has two ionizable groups, the amino group and the carboxylic acid group, and some of them have a third one on the side chain. So if you have 3 equivalence points, that can really help you to narrow down which amino acid you have, because not all amino acids have an ionizable side chain. The other thing that can also help you is looking at the pKa's, especially the pKa of the ionizable side chain. So in general, for most amino acids, you'll have one pKa right around 2, so that's this one here, and that's for the carboxyl group. The other higher pKa is for the other ionizable group that they all have, and this one is for the amino group, and that one's usually around 9 or 10. And so I will really quickly actually pull up our table of amino acids so that we can look at, my goodness, I apologize. So to look at our table of amino acids, we can see that the pKr, that indicates the pKa of the side chain. Most of our amino acids don't have an ionizable side chains. There are some that do, and they're listed down here, and they all have pretty different pKa's. So that makes it pretty easy to identify the amino acid just really based on the side chain information. The pK1, which is for the carboxyl group, and the pK2, just for the amino group, you can see are fairly consistent. There's some small differences. So if you make very accurate measurements, you can also use the pK of the carboxyl group, which is around 2 for most of the amino acids, and the pK of the amino group, which is around 9 or 10 for most amino acids. That is one really important set of information you can use. And the last thing that you can use your titration curve to measure is the molar mass of your amino acid. So when you add your titrant, your acid, or your base, you will know how much volume you added, and you can use that to figure out how many moles you added of protons, for example, to help protonate your amino acid, and you know that can help you figure out how many moles of amino acid that you have when you finally reach that equivalence point, or that pKa. So once you figure out how many moles of amino acid you have, based on how much titrant you added, you also will know during the experiment how much mass of amino acid you used to make your solution. So when you have the mass and the number of moles, you can use that, you can divide those, and you can figure out the molar mass. And as we can see in our table, why is this, sorry. As you can see from our table, the mass of each amino acid is pretty distinct. And so with those pieces of information, the number of equivalence points, the pKa's, as well as the molar mass, you can use the information that you gathered in the lab to help you figure out the identity of your unknown amino acid. So thank you for bearing with me during some of these technical difficulties, and now you are ready to complete the pre-lab questions and go into the lab. There will be a second video that talks a lot more about how to do your calculations, and you are free to watch that before the lab, or you can wait until after the lab, because this is all you really need to know to actually complete the lab itself. Great, see you in lab.